Good morning. Again, my name is Amalia Almada. I am the Associate Director of the USC Sea Grant Program based in Los Angeles. And for the past 6 years, I've co-directed the Sea Grant Deep Ocean DDT Research and Outreach Program in collaboration with Dr. Leon Gua at California Sea Grant. And today I've been asked to provide a big picture overview of legacy DDT contamination in our region, including Santa Monica Bay. I'll start with a brief history of the issue and then turn to some really interesting, hot off the press research results that looked at where DDT is today, its potential impacts on wildlife, and public health considerations. Next slide, please. So as Luke briefly mentioned, and as you all know, there are really many water quality priorities that we all think about. And I want to start with this question of why does legacy DDT still matter to our region, considering the many priorities on our plates? And we start to answer that when we look at how our region of Southern California has a particularly significant DDT contamination history. Montrose Chemical Corporation was once the world's largest DDT manufacturer, and it operated in LA County for almost 40 years. And at its peak, DDT was considered really a miracle. The inventor received a Nobel Prize. It was so effective, long-lasting, and affordable in its protection of soldiers and crops during World War II. But that ability to persist is what became the problem. We now are more than 50 years after its ban, and the DDT and breakdown products are still present in our seafloor sediments and marine food webs. And as a result of decades of DDT waste disposal in our region, the nation's largest DDT contamination sites lie off our coast. And I'll quickly explain how exactly that came to be in the next few slides. Next slide, please. So this map shows two main pathways that carry DDT contamination to the ocean. Areas in red are those locations where DDT contamination has been documented. The source is the former chemical plant, Montrose Chemical Plant, pictured with a black pin. And one major contamination pathway to the ocean was industrial wastewater, which came from the plant, traveled through the regional water system to the Joint Water Pollution Control Plant, what it was called at that time, which is indicated with the blue pin. And this contaminated wastewater was discharged through the White Point Ocean outfalls, which are pictured as purple dots. As Luke mentioned, we have a Superfund site on the Palos Verdes Shelf because of those discharges, which contain DDT contamination. The Superfund site was designated as such in the late 1990s. The second contamination pathway was through rain events, moving contamination from the plant through stormwater drainage system, primarily kind of found a pathway into the Dominguez Channel, which carried the contaminated runoff directly into the Consolidated Slip, which is an ocean inlet at the Port of LA. Next slide, please. What we now understand is that there was a third pathway, which is by direct offshore disposal of DDT waste. The manufacturing process for DDT generates really highly acidic liquid waste containing DDT and related compounds, and that waste was transported by tanker truck to the Port of LA, put into tank barges that were then towed out into the San Pedro Channel. And in this zoomed out map, you can see two historical deep ocean disposal sites that have confirmed DDT contamination. For a sense of scale, disposal site number 2 here is roughly 12 miles from the Palos Verdes Peninsula and is sitting in water approximately 1,000 meters deep. So the key takeaway across these two maps is that there are 3 distinct pathways by which DDT entered the marine environment. Again, we have the wastewater discharge through White Point outfalls, we have contaminated stormwater entering the harbor, and then direct offshore disposal from barges. Next slide, please. So what about those barrels on the sea floor? These have really dominated local news, national news. These images have become an emblem of sorts of offshore DDT contamination. Next slide, or next, please. What we now understand is that DDT was likely disposed primarily through bulk dumping rather than in the barrels. DDT is seen broadly distributed throughout the sediment of that seafloor. It doesn't appear to be more concentrated around the barrels that have been tested. There's a 1961 photograph here showing what appear to be those tanks containing DDT acid waste on a barge. So that waste would be loaded into the tanks on the barge. Barge would head offshore and liquid waste would then be pumped directly into the ocean, settle through the water column, and then accumulate in the sediment. So what is in those barrels then? Some recent research examined 5 barrels on the seafloor. 3 of them showed evidence of highly caustic waste leakage. So this is creating those white halos around the barrels. And they are seeing impact on sediment, microscopic life more than 50 years after those barrels were disposed. So although we know these iconic barrels don't have DDT, they really were a catalyst for some significant deep ocean DDT research over the past few years. And they really mobilized researchers, funders, and community members to investigate this legacy of ocean dumping in our region. Next slide, please. As offshore dumping drew major local national concern, really starting in 2020, Sea Grant became involved to really help bring people together to identify what are the most important research gaps given the expense of working in that deep ocean environment. Next, please. That momentum translated into significant state and federal research investment that was shepherded through NOAA on the federal side and the State Water Resources Control Board on the state side. And for the rest of the presentation, I'll walk through the high-level findings coming from those funded projects, what we know now and where important questions remain. Next, please. The recent research can be organized around 3 guiding questions. Next. Where is the contamination today and how is it behaving? What are we learning about potential impacts to marine life? And what does this mean for potential human exposure and health? Next slide, please. So where is the DDT today? Next, please. So I want to start really quick definition. You'll see or hear me refer to DDX. That refers to kind of the family of 6 DDT-related compounds we most commonly monitor. That includes DDT, DDE, and DDE, and each of those 3 have 2 forms. And what the most recent research tells us is that there is still a substantial amount of this legacy contamination in this deep sea floor. The latest work estimates roughly 30 to 36 tons of DDX remain in the San Pedro Basin. Most of that contamination is buried in the seafloor sediment and within a distinct layer, but that we're really talking the upper tens of centimeters of sediment. We see some, some eventual burial. There is some newer sediment that is coming on top and settling on top of this layer of DDT. A 2024 study found that much of the historical DDT dumping actually occurred before these official disposal sites were established. So you'll see these circles, disposal site 1, disposal site 2, and this map, but the disposal site boundaries we use today, including the ones on my own map in the previous slides, don't actually capture the full historical footprint because of that history of dumping. That's what you're seeing on this map, this DDX inventory. It's widespread, it's patchy in the basin, rather than really neatly concentrated in these official disposal sites. And lastly, I want to point out this pronounced peak in the DDX in this northeastern part of the basin. You see kind of an intense red area closer to the Palos Verdes Shelf. Researchers recently used the chemistry and age of the sediments across this area, the basin, and they've used that almost like a fingerprint to distinguish and identify where the DDT contamination is coming from. And they found that some of the DDT contamination in this kind of red northeastern part of the basin likely actually came from the Palos Verdes shelf instead of deep ocean disposal. So the key takeaway here is that there's a large patchy reservoir of legacy DDT in the deep seafloor from historical offshore disposal, but there may also be movement from the PV shelf into the deep basin as well in this area of the basin. Next slide, please. You can advance one more. So this leads us to what happens to DDT once it's in the sediment. Much of the DDT contamination is buried beneath a few centimeters of sediment, as I mentioned, but it can change over time. There's been a lot of research from the PV shelf finding that DDT and its breakdown products can continue to change and further break down once buried. As DDT breaks down, sometimes it can become more mobile. Can become more soluble in water. And this illustration on the slide shows the basic idea where these larger particles represent sediment, blue spaces between them represent pore water, kind of this water between these teeny tiny sediment particles. And so some DDT and its breakdown products are going to stay really stuck to the sediment, but some are more water soluble. They can dissolve into this really tiny space between sediment pores. And that's significant because this newer research is finding some of those water soluble DDT in this interstitial space in between pore water sediment cores taken from this basin. When these researchers scaled their measurements across the basin, obviously they don't cover every single part of the basin, but their estimates are that as much as 8 tons per year of these water-soluble DDT breakdown products could be moving out of this attached sediment into this pore water space. Next slide, please. Okay, so we're finding DDT can leave the sediment, go to this pore water, but can it then move into the water column above the sediment? And recently, federally funded research at Scripps provides some early evidence that it can. They installed a mooring near dump site 2 for 6 months, and they detected small but measurable amounts of DDT in that 6 meters of deep water right above the seafloor. So it's still very deep. We're talking like about 1,000 meters down. The concentrations were very low, but what's interesting is that the DDT increased closer to the seafloor. So this means, although there's not a large amount of DDT in the water, it's detectable decades after disposal, and we're seeing, that what looks like slow release as the concentrations are higher as you get closer to this contaminated seafloor. These findings are still going through peer review process, though, which is important to note. And we also don't know what these low concentrations actually mean for ecological exposure or risk in these environments. Next slide, please. Next question is whether the contamination we've been discussing is actually reaching marine life. There's been a big field of study in our region because of the PV shelf contamination. Again, researchers have detected DDT-related compounds in marine food webs across the Southern California Bight, from the very teeny tiny zooplankton to deep water fish to larger fish, seabirds, and marine mammals. More recent work has found DDT-related compounds in organisms even up to 100 kilometers from the offshore disposal area. So there's continuing research from this funded work suggesting the movement of animals and their prey is actually really important and expanding kind of the geographic range and footprint of this contamination. Another important finding from recent work is they have identified about 72 DDT-related compounds now from this area of contamination. Traditional monitoring typically focuses on 6 DDX compounds, but we now see about 72 within local sediments and species. And some of these less monitored compounds were found in wildlife at concentrations as high as or higher than the traditionally monitored DDX compounds. And importantly, like DDX and DDT, there is this pattern of higher concentrations of these compounds at higher levels of the food web, meaning it kind of accumulates and increases as you go up the food web. So the main takeaway here is DDT contamination is reaching a broad range of marine life, and this chemical mixture that they are exposed to is broader than what we've traditionally measured in the past. Next slide, please. So what we know about DDT effects on these organisms comes from decades of previous research that it can affect reproduction and development in wildlife. The newer research is asking whether some of these less studied DDT compounds can also cause biological effects. They've done some early laboratory studies, and they are finding exposure can impact survival, development, and reproduction in their, their example organisms, usually zebrafish. So this work is ongoing, it's still under peer review, but they are seeing not only compounds, these expanded lists of DDT compounds in wildlife, but we see that they're also producing biological effects in laboratory studies. Importantly, we don't know how these lab results would translate to effects in wild populations. Next slide, please. So what does this mean for people? Are people being exposed, and what is the actual risk? I'm going to focus primarily on one major pathway, which is of exposure, which is by eating fish that contain DDT. Finding DDT in a fish doesn't itself tell us the health risk. As this figure shows, exposure really depends on how much DDT is in the fish, how much fish someone eats, and how often they eat it. And that same exposure may not mean the same risk for everyone due to individual risk factors. And that's where this most recent research provides some useful context. The vast majority of recreational caught fish were below the thresholds used for California consumption guidelines. There were some important exceptions, including bottom-dwelling fish caught near highly contaminated areas. Recent research is finding that where a fish lives, where it is caught, and what it eats all helps predict how much DDT it carries. There is newer research looking at additional commercially and recreational important fish to understand how contamination relates to fish size, habitat, and movement. And state-funded study also looked at sharks and rays that aren't typically included in the fish advisories, but people do catch and eat for subsistence. And that study is showing, again, this fish movement can really connect contaminated areas with non-contaminated coastal fishing locations. So that's really important consideration in emerging research. But importantly, none of the edible mussel samples from shark and rays are exceeding advisory levels, although liver is a different story. We see much higher concentrations and exceedance of advisories in liver samples. So main takeaway here is that there is a pathway for human exposure through seafood, but contamination isn't the same as health risk. So again, it matters how much DDT in the fish, how much it's being eaten, who's being exposed. And right now, the research suggests that most recreationally caught fish are below current action levels, but we're still identifying what species, locations, and consumption patterns may affect exposure. Next slide, please.
Hi, Amalia.
So
are you able to wrap up in 2 to 3 minutes?
Yes, I'm on the end here. So I just
Thanks.
wanted to give a shout out to on kind of the policy management side that there is a lot going on right now to translate this research into public health protection. So EPA is doing a lot with fish contamination education. The PV Superfund site reached an important milestone earlier this year around its risk assessment and ongoing work on the deep ocean disposal sites to do connect federal agencies to understand the legacy of dumping at these sites. And Calaweha is updating its fish consumption guidance right now using some of the data funded by the state and NOAA. Next slide, please. So I'll skip over this because of time, but I do want to note the researchers have identified that, we answer some questions and then we raise new ones the more we dig into what the story is in the deep basin. So, there's a lot still to do to define kind of where DDT is down there. It's such a massive area in the basin. And we have a lot more to understand about kind of the scale beyond kind of these individual studies to how things are affecting at the population scale. Next slide, please. So I'll close soon, but here's a QR code if you want to learn more. The Sea Grant had put together a story map site to capture the final report from the state-funded researchers. We linked to the federal projects as well, and EPA's work on the DDT issue. And I'll close on the next slide just with a thank you and acknowledgements of the many people involved in the work represented here, our community partners, Heal the Bay, Sacred Places Institute, and SCOOS, as well as the funded researchers who have been working so hard to get this work done and out there and communicated to communities. And I'll close just with my email on the next slide for any questions. Thank you so much.